Could the human body itself eventually adapt to the red planet's harsh conditions? Evolution has equipped humans with remarkable adaptability, allowing us to inhabit nearly every environment on Earth. But Mars presents challenges beyond anything in our evolutionary history. Natural selection, the process that drives evolution, works through subtle advantages that accumulate over time. Earth's evolutionary pressures shaped our bodies for 300,000 years of human existence, fine-tuning us specifically for this planet's gravity, radiation levels, atmospheric composition, and temperature ranges. Consider the Himalayan populations who developed enhanced oxygen processing abilities at high altitudes. These adaptations include increased lung capacity, altered blood chemistry, and more efficient oxygen transport systems. After approximately 3,000 years of natural selection, Tibetans can thrive in environments with oxygen levels 40% lower than sea level. Similarly, Andean populations developed larger chest capacities and higher red blood cell counts to cope with their mountainous habitat. Mars, however, presents simultaneous challenges far more extreme than Earth's harshest environments. The adaptation gap becomes clear when comparing Earth's high-altitude regions to Mars conditions. While Tibetans adapted to oxygen at 60% of sea level concentrations, Mars' atmosphere contains less than 1% of Earth's pressure with virtually no oxygen. This represents a fundamental difference rather than a more extreme version of the same challenge. What makes Mars particularly challenging is that many of its conditions present binary survival thresholds rather than gradients along which adaptation can occur incrementally. The human respiratory system requires a minimum partial pressure of oxygen to function. Below certain thresholds, oxygen simply cannot transfer to the bloodstream regardless of evolutionary adaptations. The pressure gradient necessary for respiration is a physical requirement based on gas exchange principles. Similarly, the absence of a global magnetic field on Mars means surface exposure to radiation levels that can damage DNA and increase cancer risk substantially. Natural selection might eventually favor individuals with slightly better DNA repair mechanisms, but the protection would be marginal compared to the magnitude of the threat. Humans have evolved on Earth for hundreds of thousands of years, yet we still haven't developed natural defenses against many Earth-based radiation sources. In reduced gravity, over many generations, natural selection might favor individuals whose bones and muscles maintain better structural integrity with less gravitational stress. The musculoskeletal system shows remarkable plasticity, responding to different loading conditions throughout life. However, these adaptations represent optimizations within Earth's environmental range, not fundamental rewiring of human physiology. Artificial selection, deliberately selecting for advantageous traits, could potentially accelerate adaptation. This approach involves identifying individuals with characteristics beneficial for Mars conditions and encouraging reproduction. However, this raises profound ethical questions and practical limitations. The effectiveness of artificial selection depends on existing genetic diversity, and many traits critical for Mars survival likely don't exist in the current human gene pool. The limitations of natural adaptation don't mean that humans couldn't survive on Mars. They indicate that technology, rather than biology, must bridge the gap. Habitats, spacesuits, and other protective systems would remain necessary regardless of how long humans live there. Natural adaptation might eventually help humans better tolerate the 38% gravity, but it wouldn't eliminate the need for radiation shielding or pressurized environments. If humans established a permanent, self-sustaining colony on Mars with limited return to Earth, genetic drift would likely begin to create a distinct population over generations. Initially, these differences would be subtle, perhaps slightly different bone structures or cardiovascular systems optimized for lower gravity. Over thousands of years, assuming continued isolation, these differences could accumulate until Mars-born humans might struggle to readapt to Earth conditions. This potential divergence raises profound questions about human identity and the possibility of speciation as we spread throughout the solar system. Would Mars colonists eventually evolve into a separate species, given enough time and isolation? The possibility exists, though the time frame extends far beyond our current planning horizons. The immediate solution to Mars' environmental challenges lies not in waiting for natural adaptation, but in directly intervening. In human biology, genetic engineering technologies like CRISPR offer theoretical pathways to enhance human capabilities beyond what natural selection could achieve. These approaches could potentially target specific adaptations needed for Mars: radiation resistance, improved bone density maintenance, enhanced oxygen utilization, and modified cardiovascular function for the lower gravity environment. However, significant technical and ethical barriers still stand before such modifications could be safely implemented. The long-term consequences of genetic alterations remain largely unknown, and unexpected effects could emerge over generations. The complexity of the human genome means that modifications intended to address one challenge might create vulnerabilities in other systems. The gap between what we can imagine and what our biology permits represents the fundamental challenge of becoming a multi-planetary species. Technologies that protect humans from Mars' environment will remain essential indefinitely, underscoring the necessity of engineering solutions rather than biological ones. Engineering humans for Mars. Technology may shield our bodies from Mars hostility, but what if our bodies themselves could become the technology? The revolutionary gene editing tool CRISPR, Cas9, represents a paradigm shift in our approach to Mars colonization. Unlike previous sections where we explored how to protect humans from the Martian environment, genetic engineering invites us to consider a more fundamental question. Could we redesign ourselves to thrive where evolution never prepared us to survive? CRISPR, Cas9, has transformed genetic modification from science fiction into scientific reality. This molecular scalpel allows precise alterations to the genome, targeting specific DNA sequences with unprecedented accuracy. Such precision opens fascinating possibilities for enhancing human capabilities specifically for Mars conditions. This power comes with significant limitations. Among the most concerning are off-target effects, where CRISPR machinery unintentionally modifies genes other than those targeted. These unplanned edits could trigger cascading health consequences that might not become apparent for generations. These modifications would potentially create genetic changes that pass to descendants, with all the unforeseen complications that might entail. Radiation resistance represents one of the most promising applications of genetic engineering for Mars adaptation. Radiation poses an existential threat to Mars settlers, but certain organisms on Earth have evolved remarkable resilience. The bacterium Deinococcus radiodurans stands as nature's supreme radiation survivor, capable of withstanding radiation doses 1,000 times what would kill a human. Scientists are investigating the genetic basis of this extraordinary resilience. Deinococcus radiodurans possesses highly efficient DNA repair mechanisms that allow it to restore damaged genetic material with remarkable efficiency. In theory, we could incorporate genes responsible for these repair mechanisms into human cells, enhancing our ability to withstand constant radiation bombardment on Mars. Imagine a Mars settler like Elena, born in 2050, working outside her habitat for hours without concern about cumulative radiation damage. Her cells continuously repair radiation-induced DNA breaks, similar to how self-healing materials automatically fix microcracks. The implications extend beyond space travel. Such modifications could reduce cancer risk and slow aging processes even on Earth. Another critical challenge is maintaining bone density in Mars' reduced gravity. On Earth, our skeleton constantly rebuilds itself in response to gravitational stress, but this process falters in low-gravity environments. Genetic engineering could intervene by modifying genes involved in bone formation and maintenance. Scientists are researching ways to enhance the activity of genes promoting osteoblast function, cells responsible for building new bone tissue, while suppressing genes triggering bone breakdown. This balance could be adjusted to favor bone preservation even when gravitational signals diminish. The objective would be creating a modified bone maintenance system that remains robust despite receiving weaker gravitational cues, much like upgrading a car suspension system to perform optimally on different terrain. Oxygen utilization represents another frontier for potential genetic enhancement. While we can't change Mars' thin carbon dioxide atmosphere, we might improve how efficiently our bodies use limited oxygen available in artificial habitats. Genetic modifications could enhance oxygen uptake, improve transport through the bloodstream, and optimize cellular utilization. This might involve modifications to hemoglobin structure to increase oxygen binding capacity or enhancement of mitochondrial efficiency. Such changes would be crucial in environments where atmospheric resources must be carefully managed, comparable to upgrading an engine to run efficiently on limited fuel. Synthetic biology takes genetic engineering further by designing entirely new biological pathways specifically adapted for Martian conditions. This could involve engineering organisms with metabolic pathways that don't exist in nature. Settlers might have enhanced abilities to produce essential vitamins their bodies cannot naturally synthesize, reducing dependency on nutritional supplements. Engineered symbiotic relationships with microorganisms could help process Martian resources. Synthetic biology might even create organisms capable of transforming Martian atmospheric components into breathable air or useful compounds, effectively turning biology into infrastructure. However, the time frame for developing these technologies exceeds current Mars mission planning windows. While CRISPR has advanced rapidly, safely engineering complex traits in humans remains in its infancy. Current research primarily focuses on correcting single gene disorders, a far simpler challenge than engineering complex adaptive traits for extraterrestrial environments. Developing safe genetic modifications would require extensive research phases spanning decades. The most optimistic estimates suggest preliminary human applications might begin in the 2040s, likely limited to therapeutic rather than enhancement purposes. The ethical considerations surrounding human genetic engineering add another layer of complexity. Society must confront difficult questions about consent, equity, and the very definition of humanity. Who decides which modifications are permissible? Would genetically enhanced Mars settlers constitute a new branch of humanity? Modifications that provide advantages on Mars might create disadvantages if individuals or their descendants return to Earth. The human genome represents an intricate system developed through millions of years of evolution. Alterations made for specific environments could ripple through this system unpredictably. How would genetically modified individuals view themselves and relate to unmodified humans? Will we create a two-tiered species, potentially leading to discrimination? These questions extend far beyond technical feasibility into ethics, philosophy, and human rights. Despite these challenges, genetic engineering remains one of the most promising long-term approaches to enabling sustainable human presence beyond Earth. Unlike mechanical life support systems that can fail catastrophically, genetic adaptations would become intrinsic to the organism, providing continuous protection without external power requirements or maintenance needs. Throughout our exploration of space, we've often imagined conquering other worlds. But as we've seen, the reality is more nuanced. Our bodies, these remarkable machines, evolved over millions of years on Earth, face profound challenges beyond our home planet. The path forward requires harmony between our biology and alien environments. This means developing technologies that complement our evolutionary heritage, advanced shielding, closed-loop life support systems, and perhaps careful genetic modifications. Our success depends on this delicate equilibrium. The question becomes about maintaining our essential humanity while adapting to worlds our ancestors never imagined.